Hello, everyone, and welcome to the sixth in our Environmental Epigenetics Symposium webinar series. We're delighted to have Janine LaSalle and Keith Dunaway from UC Davis speaking today. If you are interested in seeing any of our 5 previous webinars, please look up germlineexposures.org. They're on the homepage. Also at asfpodcast.org, and you can also just do a search on the Autism Speaks website. My name is, if I can click on this, my name is Jill Escher. I am one of your hosts today, along with Alicia Halliday, Chief Science Officer from the Autism Science Foundation, Matt Pletcher, VP for Genomics at Autism Speaks, and our other host today is the Autism Brain Net. You can register to participate in BrainNet and just find out more information at takesbrains.org. Few logistics before we start. If you have questions for either of the panelists, we'll take questions after Dr. LaSalle. Oops, that doesn't work. After Dr. LaSalle speaks, and then again at the end of the webinar. You can type those into the control panel. There's a question bar, and those will show up typed, and we will moderate them along the way. All of the participants, except the speakers, are on mute. Sometimes your screen is hiding beneath your browser, so if you can't see the screen right now, take a look under there. The webinar is being recorded. This, we think, will go to maybe about an hour and a half. So a lot of people can't stay that long, but fear not, because you will receive a follow-up email with the link to the archive. With that, I turn it over to Dr. Matt Pletcher.
Great. Thank you so much, Jill. And again, we really thank Dr. LaSalle and her colleague, Keith Dunaway, for joining us today. As a point of introduction, Dr. LaSalle really is a pioneering researcher in the field of epigenetics and its role in autism. Dr. LaSalle got her start by studying biology as an undergraduate at Randolph-Macon College. From there, she went on to earn a PhD in immunology at Harvard. She moved on from there to join the Howard Hughes Medical Institute and Boston Children's Hospital as a postdoctoral fellow with a focus on human genetics. After completing her postdoc, Dr. LaSalle established her own lab at UC Davis. She is currently a professor of medical microbiology and immunology and the chair of the Integrative Genetics and Genomics Graduate Group at the University of California, Davis, as well as a member of the school's really world-renowned Mind Institute. Her laboratory focuses on epigenetics of neurodevelopmental disorders with a special focus on autism and related disorders through the investigation of genetic mouse models, neuronal cultures, and postmortem human brain. And today we get to hear a little bit more about some of the work that Dr. LaSalle is doing. So from there, I'll turn it over to you. And again, thank you so much for joining us today.
Great, thank you. So, show my screen. Okay, are we seeing the screen? Hope so.
Yes, you just want to make that.
Yes.
Widescreen. Yeah, there we go.
We see it. Yeah, you will want to put it in full play mode.
Okay.
Perfect.
Yeah.
Great. Okay, well, thank you very much, Matt, for that introduction, and thanks to Jill and Alicia for inviting us to do this. It's a great opportunity for us to share our work with a broader audience. So I'm going to start out with an introduction to epigenetics, gene-environment interactions, and give you a little past history of some of the work we've done previously looking at single gene autisms and gene environment interactions. And then I'm going to let Keith Dunaway take over to talk about our recent publication that he headed up. So this is a slide I'd like to start with because this is kind of the focus of our lab. We're kind of at that interface of autism and epigenetics. And I like to use the analogy of these Pandora boxes on either side because it's almost like there are so many, epigenetics is a fairly new field and there's a lot more questions and answers, kind of similar to the field of autism. And so again, in this sort of big open, there are a lot more questions and answers, and our contribution is really trying to use some of the more recent newer technologies such as next generation sequencing to get a handle on what is the connection between epigenetics and autism. But historically, I like to say that really it was genetics that pointed us to the epigenetics. And a lot of the children with more rare syndromic forms of autism um, um, are what got us interested in this field to begin with. So I actually did my postdoctoral training on 15q and Prader-Willi-Angelman syndrome, which since then has been led to the duplication of chromosome 15 that you'll be hearing a lot more about today in Keith Dunaway's portion. We also work on Rett syndrome, which is, again, I think kind of what I like to call the Rosetta Stone of neurodevelopmental disorders, because this gene called MECP2 really pointed out that chromatin and DNA methylation was important in the developing brain. In addition to the ones that my lab works on, which is on this side of the slide, I do want to kind of mention a few others because the epigenetics, even for these classically genetic syndromes like Fragile X, which is a repeat in the FMR1 gene that causes the disorders, epigenetics serves to actually turn off that copy of that gene in boys. And even Down syndrome is, classically considered a genetic disorder of having a whole extra chromosome 21. There's definitely some compelling evidence for epigenetic alterations in Down syndrome as well. And then Williams syndrome also a large deletion region that includes some evidence for chromatin factors. But again, I think what's interesting and what's come out of the field of genetics and genomics in autism is that disorders like Rett syndrome are probably just the tip of the iceberg. And of sort of unbiased sequencing approaches, functions involved in chromatin are kind of the #1 hit for genes that are syndromic or found to be responsible for autism. So here's another analogy I like to make, which is one of an apple tree. So if you think about the things that were the obvious ones to look at first, the low-hanging fruit, the genes that act at synapses, these were sort of a lot of candidate approaches or maybe found by syndromic disorders. But when sort of the advent of genomic sequencing and exome sequencing, a lot more of what chromatin genes were sort of unearthed in this process. So obviously I mentioned MECP2 as one of these, but there are many other methyl binding proteins, methyl CpG binding proteins that act at this. So again, the analogy here of the tree, I won't focus on what all these genes do, but you can think about it just like a healthy apple tree and with great fruit. What's going on underground in the root system is really important. So you can think about chromatin factors, which are basically in the nuclei of neurons, as like the roots of healthy synapses. And so genetically, you can alter the roots by sort of rare mutations in a lot of these genes. Epigenetically, we'll talk about how you can alter the roots of these little red dots of DNA methylation and the histone modifications that can occur down here. But also I want to point out that it's not just genetics that affects the health of an apple tree. Anyone who's done any farming knows that the environment makes a big difference, right? So things like diet, so if we bring this back to humans, just like rain and sun conditions, things like that can really affect the health of the apples. For humans, things like diet, toxins in the environment, stress, metabolism, inflammation, all of these can feed back on the roots of the tree to affect the health of the synapses in the brain. So from that, so what do we know about the gene-environment interactions? And again, this is where it gets a little challenging. It's nice to think that, I think the easy way to think about gene-environment is they're in separate boxes that don't interact. But the reality is it's not a simple math equation. You can't just add genes plus environment because there are so many complex interactions. And again, bringing back to the analogy of the tree, same way. It's you can have trees that are genetically stronger in drought conditions than high rain, and ones that love the rain and not the drought. So in the same way, you get these complex interactions. So here's another analogy of a Rubik's Cube. So again, I think this is kind of what, I like this picture because it's kind of what it feels like when you're down in the trenches working on these gene-environment interactions. They're complex, and every time you think you have one piece of the puzzle solved, there's another monkey wrench thrown into the whole system. So I'll kind of give you a little glimpse of what we've seen of that in our experiments. So thinking about gene-environment interactions, again, it's really a two-way street. You can't think of them as completely independent from each other. So on the one side of the street, we've got genetic differences that can influence environmental susceptibility. So there are genes in your genome that can affect how you metabolize toxins, how you metabolize dietary compounds, what genes that affect your microbiome, and all sorts of things. On the other side, you've got environmental influences that can influence gene expression. So all of those genes in your genome can be impacted by epigenetic changes. And that's what we'll be kind of focusing on today. But last but not least, you can also have a direct impact of the environment on the DNA. There are some, not the factors we'll be talking about today, perhaps, but definitely there are plenty of mutagens out there where the environment can actually be causing some of the genetic changes. Or again, I think what we're kind of getting at in some of our research is you could have epigenetic changes through an environmental factor that could then lead to damaging the DNA or genome instability. So again, I just like to reinforce the idea that as great, I'm a human geneticist. I love staring at A, T, Gs, and Cs in the sequence. But the reality is that the sequence of your DNA, your whole genome, is not in a lockbox away from other influences on the body or your cells. And the whole field of epigenomics and integrative genomics is really looking at DNA as part of an integrative part of the living cells that's affected by all the things that are affecting the brain, the neurons, all affect all of these intricate things going on inside the nucleus. I also want to bring in, this is, it's actually getting a bit old now. This is back in 2010, but it was actually the first time that epigenetics made mass media, made the cover of Time. But of course, it was a kind of headline that I kind of take a little issue with because it said why your DNA isn't your destiny. And it kind of made it sound like this new science of epigenetics is this radical anti-genetics movement. And I just want to stress, and I'll show you, examples of this, that epigenetics literally means on top of genetics. It doesn't mean that genetics is not important. It means that there's layers on top of your genetic code that are really important. So what is epigenetics? And this is kind of my teaching slide for this. First, it's good to define what is epigenetics. So they're basically inherited and reversible modifications to nucleotides or chromosomes that do not change the sequence, but can alter gene expression. And I like to think of it as multiple layers on top of your DNA code, starting from the one that we'll be focusing on mostly today, which is DNA methylation, which is simply really just a methyl group added to cytosine residues in the genome. The second layer is what's done to histones. So the histones are these little tails that stick out of the, so you can think about your DNA inside the nucleus. It's not just a big bag of spaghetti. There's order and organization. And the histones are basically the spools around which your DNA is wrapped. And these spools have a lot of different changes, sort of flavors on them that can modify how tightly the DNA is packaged up inside the nucleus. The next layer on top of that, again, is order and organization. In order to get a gene expressed at the right time and right place, you got to get it to the right place. So having these chromatin loops is a way of, you can think about it as sort of macrame of your DNA to kind of fit it into the nucleus and get that gene in the place at the right time. And then you can think higher up from that. Again, we like to think linearly in chromosomes, but inside the cell, all of this DNA is in three-dimensional space, and it's still organized in chromosome units, even when they don't look like chromosomes. This is an interphase nucleus where each chromosome has been painted by a different color. And so you can tell that the active genes tend to be out in between these spaces, where the inactive genes are sort of in the interior of these spaces. So there's order and organization to your DNA, even inside every living cell in your body. And for genes, especially genes that are involved in neuronal development and neurons specifically, location does really matter. It's kind of like real estate. And last but not least, and this is an area I won't be talking about, but it's a fascinating emerging area, is the whole area of regulatory RNAs. And these mean that not all genes in your genome encode proteins. There's a lot of small RNAs and large RNAs that are acting sort of enzymatically on all these other layers of epigenetics. So these give specificity to some of these other epigenetic layers. And so I'm just going to focus primarily on the DNA methylation today, and that's really the topic of the project that Keith will be talking about. So here's another way of looking at methylation. For hopefully some of you guys have had organic chemistry and you remember the 5-carbon ring. Well, this is really just a methyl group added on to a cytosine residue at this 5 position. So that's what we mean by 5-methylcytosine. But imagine this molecule in a lot of more throughout your whole genome. So in the whole genome, there's about 28 million of these CpG sites. So by CpG, we mean that C is 5' of a G in the DNA sequence, and the methyl group, and this is the one that's really highly methylated in the mammalian genome. And you can see by this diagram that it's on both strands, and the cool thing about CpG sites is they're mirror images of each other in the DNA, double-stranded DNA, so you get a methyl group on each strand, and that's why it is really highly maintained. And the human genome is one of the is the most highly methylated genome of all species, that we've been acquiring more methylation with evolutionary changes. But again, the expense of having all of these CpGs to methylate is it takes a lot of methyl groups, right? And so I just kind of want to point out this kind of sink analogy here. Why, and you'll see that what we're building to is really that, you know, there's not a single cause of autism. There's a lot of genes, there's a lot of environmental factors that could be involved, but they all may be acting on a similar type of pathway. So genetic factors, large chromosome duplications that you'll see in our study, as well as combined with environmental factors, could be impacting this sink, this big need we got to methylate these 28 million CpG sites in every cell in the body. One of the major sources, there's a lot of things that can drain the sink, but not that many that can supply it. And the major supply group is from, I mean, we definitely recycle methyl groups in the body, but the major new supply is from your diet. And that's why folate, B vitamins, taking prenatal vitamins before conception and pregnancy is really critical for providing those methyl groups. So just to say that you can have these different outcomes. Again, if you're really lacking a lot of this, the fetus isn't going to even implant, it's not going to survive. But subtle changes to the size of the sink could be subtly affecting epigenetics. And again, the combination of these factors, one thing may not be enough on its own, but a combination of too much risk and not enough supply. And so the evidence kind of supporting this general model from the genetics field is that you know, there are these large copy number variations that are often associated with autism, but a lot of times it really is more the global burden. And this is work by Santosh Girajan. And then they just had a recent paper collaborating with our group here at Davis showing that the combination of too many chromosome duplications plus air pollution increased the risk up to odds ratio of 3.4, and that was much more than either risk factor alone, which was down in the 1.3 to 1.8 range. So again, the combinatorial effect of both genetic and environmental factors may be affecting the sink. And again, that's probably why preconception use of prenatal vitamins is protective for a number of different disorders, including autism, Down syndrome, surprisingly. Again, that's a whole extra chromosome 21, as well as neural tube defects, which is the reason why it's currently prescribed for all women. And so this is my attempt to try to plot some of the gene-environment interactions that could be interfacing with DNA methylation and autism here. So again, as I was saying, multiple factors may involve. I mean, there's certainly the syndromic forms of autism would be a single gene or a single copy number variation may be enough to be causal on its own. But the reality is most forms of autism are not explained by single factors, and that there could be multiple sort of risk factors going on, counteracted perhaps by some protective factors. And again, there's a lot less known about protective factors. We do know that being female seems to be protective both from the genetic load, particularly than being male. So I have that up there under the genetics. And then taking prenatal vitamins periconceptually is the one that's known to be protective for autism. Although timing of this is really critical. It needs to be done right around the time of conception in that first month of pregnancy. There's a growing list of environmental factors that have some degree of increased risk. So the two up here are pretty rare in the population. If you get rubella infection during pregnancy or thalidomide, it really increases the the risk for autism, but most of these are more in the sort of between 1 and 2 odds ratio range, and that's increased maternal age, increased paternal age, increased grandparental age. An interesting study that just came out that the Escher Fund funded on grandmaternal smoking it increases the risk of the grandchild having autism. Maternal obesity during pregnancy is a risk factor. Inflammation, this could be from asthma, infections during pregnancy, also increase the risk. And then things like proximity to freeways, which is a proxy for air pollution, as well as living close to pesticide exposures in agricultural areas. So again, the idea is that probably none of these alone is enough to be completely causal for autism, but together with some genetic risk and not having enough of the protective factors may impact the total risk. So now I'm going to talk to you about this class of persistent organic pollutants. These are compounds that we got into a number of years ago when again, I'm trained as a geneticist, but I asked the toxicologists and the epidemiologists about factors that they think could be potentially involved in autism. And this one is definitely a culprit for neurodevelopmental disorders anyway, that's been known for many years. So this is what the chemical structure looks like. There's actually a whole bunch of classes that depend on different numbers of these chlorine molecules here. And these were used heavily from 1950 to 1976 when they were banned as flame retardants, insulation in a lot of industrial chemicals. But the reality is they're still being found in a lot of places. There's the caulking in old schools was loaded with these PCBs, so they're still coming out in schools that were built in this time period. A lot of these are being found in New England. I think now even there's some of the newer colored paints have some of the different congeners of PCBs, but they're a byproduct of some of these really brightly colored paints that are around. And the problem with these, and even if, again, if they've predominantly been, banned for a long time ago, but they bioaccumulate in lipid-rich tissue, so they tend to bioaccumulate up the food chain. So even if they're in very low parts in like, the ground and the ocean and our environment, as animals, eat up the food chain, so something like a sea lion, what's been eating a lot of fish that may have a lot of PCBs that have accumulated, they're going to have much higher levels. And I hope you also realize that, we're not sea lions, but humans are also at the top of the food chain. So the milk, the meat, anything, with this lipid-rich, and particularly fish as well, you can be getting high levels of PCBs even today. And before our studies, there actually had been some evidence. So in Greenlandic Inuits, populations where their diet is completely composed of fish, they have particularly high PCB levels, and that was associated with reduced DNA methylation in peripheral blood. So there was some potential evidence that epigenetics could be involved in that process. So another class of persistent organic pollutants that I'll be mentioning are these PBDEs. And unlike PCBs, so PCBs were kind of along with DDTs. You may have also heard of another class of persistent organic pollutants. They're used as an insecticide. That use of that was definitely decreased, banned around the same time as PCBs. So even though these have declined in the environment, we're still finding evidence in lipid from animals and humans. But PBDEs were sort of the replacement chemical to take the place of the PCBs. And that you can see the structure looks pretty similar, but they're bromine molecules instead of chlorine. And these have really increased in use. They're used, again, as flame retardants, so pretty much anything they don't want to catch on fire, which includes all your electronic devices, textiles, bedding, couches, has really risen in the last several decades. And they can also, both of these chemicals can also act as steroid hormones, which have known adverse effects on neurodevelopment. So I'm going to just briefly touch on, these are two studies that got us into the field of environmental epigenetics and autism. And that was a few years ago, but I just want to bring up kind of our basic conclusions. And then this was the study that really led to the one that Keith is going to talk about, which is this connection between DUP15Q syndrome and PCB95. So we originally started to this, we were both looking at a mouse model where we took a candidate approach of let's look at a mouse model that's known to have social deficits, which is an MECP2, a Rett syndrome model, a mutation in the MECP2 gene, and look at the effect of the addition of PBDE exposure. And this was one where we took sort of a pan approach to looking at postmortem brains. And again, because these persistent organic pollutants are very lipophilic, and brain has a lot of lipids, they can really, what you have in postmortem brain samples is reflective of almost your lifetime accumulation of these chemicals. So here we took sort of an unbiased look and said, what kind of congeners of both PCBs and PBDEs are we finding in the brain? So first, let me talk about this mouse study with MECP2. So again, this is a mouse model of Rett syndrome. It's actually a mutation found in Rett syndrome. It's a 308 mutation that's a truncating mutation. And what we did was actually, the way you breed these mice, because it's an X-linked disorder, the mother or the dam has the mutation. We expose the dam for about a 10-week period, which we call perinatal, because it includes 4 weeks prior, 3 weeks of in utero life, and then 3 weeks when they're actually, when the pups are being, there's lactation, so that the mom is still exposing the pups through her milk. And we chose initially 2 different doses based on what was in the literature, as well as a vehicle control. And basically our vehicle for this was actually corn oil on cornflakes. So because we have to dissolve it in an oil, it's lipophilic, and then put it on a cornflake, and the mouse, every day you have to watch the mouse eat the cornflake. And we did this for 10 weeks. It was a lesson in patience. In the end, we should have ended up with 12 different treatment groups based on the different dosage. And I'll tell you, we ended up just going with the lower dose based on a reproductive effect we saw in the higher dose. But because this is an X-linked disorder, we look at both males and females. So females are heterozygous for MECP2 mutation, and more like the Rett girls in humans, where males are hemizygous. They only have 1 copy of MECP2, and that's the mutant copy. And so the idea behind this experiment is we were going to look at for long-lived effects on social and cognitive behaviors, and then epigenetic changes that could be a result of these long-lived effects. So yeah, I just want to point out that we ended up dropping, I mean, at the higher dose, we almost got no pups. Only 50% of the litters survived to postnatal day 8. Yeah, so we lost so many pups with the high dose. It was an interesting, we didn't design it to be a fertility experiment, but we definitely learned. So that's kind of interesting in itself, that having both the genetic risk and an environmental exposure to high dose was enough to cause a major reproductive effect. And interestingly, when this same dose is used in wild-type mice, you don't see that fecundity effect until, or reproductive effect until 10x higher dose, 10 times higher dose. And this slide right here shows you what the dose looked like in terms of where we were. So this is the human brain that I'll talk about. It was done in the other study. So the average sort of level of this BDE47 that we were giving the mice was right about here. The high dose was definitely higher than most of the humans. There are a couple out here in the higher range. So the low-dose mouse that I'll be showing you the behavior for was really right about the average level of what we were seeing in human postmortem brain. And we saw effects even just on the weight, the pre-weaning weight. So this is the weight right before the early life of a mouse. And you can see that the ones that even had the low dose were smaller than their littermate control, or the controls that had the wild type controls, which are these plus marks right here. Okay, this slide is a little complicated. I just want to give you the bottom line, which is we looked at a lot of behaviors. So the behaviors are all here on the left, and these behaviors look at a number of different things, including anxiety, sociability, social preference, locomotion, learning and memory, and social interaction. And then because of the complexity of this gene-environment interaction, we did the statistics to look at either the environmental effect, which is that on the BDE47 exposure, the genetic effect, or the interaction between the two, which is this column. And again, keep in mind that the dosing ended here. So these are all behaviors below the ultrasonic vocalizations. These are all behaviors that are sort of long-lived effects by having that dosing in really early life. So quickly, the BDE47, the environmental effect, was seen to be stronger in the females than the males, whereas the genetic effect was stronger in the males than the females. And this is expected because, again, remember that the males only have one copy of MECP2, and that's the mutant copy, where the females are heterozygous. But interestingly, the interaction effects were also specific to the females and specific to a couple of behaviors. And I'll just briefly show you some examples of that. So the Morris water maze is the classic test of learning and memory for mice. You put the mouse in a swimming pool, and mice don't like to be wet for very long, so they're motivated to get out of the swimming pool, to get to drier grounds or to platforms. So you put a platform in there, and over 4 days of testing, what happens is it takes them a longer time on the first day of testing to find the platform, but they learn over time to swim there faster. Okay, so if you look at the wild type mice, you can see they're learning. But in the mutant mice, and particularly the mutant mice that also had BDE47 exposures, they really didn't learn at all. And here's an example of a trace of one of those mutant mice. And instead of, a wild type mouse that's learning pretty quickly, quick path to the platform, this mouse is just sort of swimming around and not ever making it to the platform. And so they're sort of a flat line across. They really just didn't learn. We control for things like swim speed, so that's an example here. There was no significant difference in their swimming speed, so it didn't seem to be a motor deficit. It was a learning deficit. So this was showing a compounding effect of both the genetics and the environmental factor. We also looked at a couple of social behaviors. This is just ultrasonic vocalization. So mice, they don't have language, but they do make vocalizations that can be picked up by ultrasonic sensors. And this is a way that the pups cry out to find, so the mother, the dam, can go find them. So it's really a test of both communication as well as anxiety, because the pups are crying out to be found, so they're not left out in the cold. And you can see that there was a significant decrease in the ones that were exposed, and this was actually independent of the genotype. So this was an environmental effect of the BDE47, and that was similar to the second test we did of sociability. So this is Jackie Crawley's classic 3-chamber slide test, where you put a mouse, there are 2 chambers on either side of this 3-chamber apparatus. You put the mouse in the center, and it can choose to either spend time with a novel mouse or an empty cage. And you can see that the time that it's spent interacting with the social, in the social area, which is around the mouse, was lower in the mice that were treated by with this environmental factor BDE47. And again, this was independent of the genotype and was only seen in the female mice. And interestingly, we also, Keith is going to tell you a lot more about methylation and some of our newer methods of looking at it. But for this time, we were using methylation dot blot, which is really just a global measure of how much total DNA methylation is in the brain. And interestingly, we saw a significant decrease in that correlated with that decrease in the sociability, meaning that the female mice that had lower, that were treated with the BDE47 environmental chemical had lower amounts of DNA methylation. So now I'm going to tell you about the second study as sort of a foray into what Keith is going to tell you about. And this was the human brain study. So here we wanted to take, we wanted to look across human brain samples. So again, these are often somewhat hard to come by, so our ends were fairly small, but this was over 100 brain samples. And within that, we had 3 groups. We wanted to obviously look at idiopathic autism, because our hypothesis going into this, maybe somewhat naively, was that it was more likely that the idiopathic autism, where we don't know the genetic cause, may have environmental exposures. But as a control group, we included several genetic causes of autism. And again, because these are rare syndromes, we didn't have these kind of numbers. But so we combined several different genetic disorders, including Rett syndrome, Prader-Willi, Angelman, and Dup15q syndrome, as well as Down syndrome. And I guess the big surprise that we had, well, one, that we saw a bigger effect with PCBs than PBDEs. And again, these are the newer exposures, and these are the older exposures. But it seemed to be specific to this congenitor called PCB95. And the real significant group was this genetic group that was higher on the PCB95. And that led us to a number of questions. And one of those, again, because there's sort of a timing of exposure, we actually went back to the brain bank data and had to kind of calculate back the year of birth because we thought that was an important variable to control for. And you can see that most of our samples are kind of more recent years. But interestingly, this genetic effect that we were seeing, a significant effect, was due to really two categories of the syndromic disorders. Predominantly due 15q syndrome, because 5 out of 6 of those samples showed higher PCB95. But then we also saw some Prader-Willi samples. But you can see the Prader-Willi were born, they've been in storage for a while, and they were born back in the 40s to 50s. And that was only 3 out of 6, and you can see we had a lot fewer controls. So we really then decided to focus on this Duke 15q syndrome because it was pretty striking and unexpected association between this syndrome and exposure to PCB95. And it did raise some interesting questions about this, is because all of these samples were from children born after the ban. We were thinking that perhaps they were getting them from their mothers, and that's similar to, they get the duplication of chromosome 15 from their mothers as well. So it was a question of, which comes first. We did in this study do a preliminary look at DNA methylation by a line 1 pyrosequencing assay and saw global hypomethylation. And then we published this back in 2012. But basically what I'm going to do is stop here. This is where Keith Dunaway's story is going to take over, asking the question, where in the whole genome is this global hypomethylation, and what sense can we make out of this interaction between Dup15q syndrome and PCB95? So with that, I think I'll stop and take questions on my part, and then I'll introduce Keith to do the second part.
Great. Thank you very much, Jean. So I think just to remind folks on the phone, if you're interested in asking a question, on the right-hand side of your screen, likely in the toolbar for the WebEx, there's a box that says questions. Type in your question there, and we can ask Dr. LaSalle your question. While we wait to see if any folks want to pose questions, I believe Jill, you had a couple questions you wanted to use to get started.
Sure. Yeah. Thank you. Gosh, I could go on all day about these concepts and this really incredibly fascinating research. But I wanted to ask one that was just kind of more conceptual in nature. And I think Dr. LaSalle, of my interest in exposures on the germline, really on the foundational biological program, as opposed to exposures to the somatic DNA. That would be, DNA in the actual brain or in the central nervous system, particularly during development, early development. And this is, again, a conceptual, not a precise biological question, but one thing I run up against when I discuss with researchers some of these concepts is a kind of conflating of gene-environment interaction. That is, they conflate it, I think, with a single idea of exposures on the developing somatic cells without sort of sequestering as a separate biological concept exposures to the early germ cells, which would be the progenitors of the egg and sperm, and the very dramatically different biological or molecular biological effects exposures can have on those two very separate phases of the life cycle. And I'm wondering, it doesn't directly have to do with your research, but I'm wondering if you can speak to that a little bit, and if there's any thinking in autism research about those as very separate categories of exposure.
Yeah, I think it's important because, and again, I think with this mouse study that we did, yeah, it was a 4-year grant. So I originally wanted to do some transgeneration because I think what Jill Escher's getting at is this issue that when you expose a mother, in this case a dam of a mouse, to an exposure, you're not just exposing her and her offspring, but the oocytes of their gametes of the next generation. So we were not able to look at a transgenerational effect in this study, but I think that would be very interesting in the future. But yeah, I think there are two things. Yeah, so, and it's fascinating about chemicals too, because I mean, the advent of man-made chemicals have been pretty much since World War II, and we've gone through a few generations, a couple generations since then. And sometimes I think, yeah, so as much as I think it's good to, when you find a chemical has bad effects on brain development, like PCBs, you should ban them. The challenge is we keep changing the chemicals. So when PCB was banned, you bring in PBDE. And I think as an epigeneticist, I really think about these changes, the epigenetic changes are evolutionarily adaptive. So because it's hard to change your genome in a changing environment, but you can change your methylome, you can change your epigenome more quickly. But if we keep purposely changing our environment so much, it gets a challenge even for the epigenetics to keep up. So, I'm not advocating for anything. It's just as perspective as thinking about things in light of evolution and our adaptation to change, that we're constantly changing policies for medicines, for chemicals, for everything, and it means that our bodies are working hard to keep up with those changes.
Yeah, well, that's an interesting perspective. I had never really thought of it in that way. But thanks for mentioning the grandmaternal smoking study that was just published.
Yeah, that's great. I just read that.
Yeah, I
Very
appreciate
intriguing.
that.
And that's
But
nice
it's nice to get something out the door.
If the mothers didn't smoke, there was a bigger effect. It was interesting.
I was about to mention that, yes.
That's fascinating. So again, it's the changing environment thing that may be some of the problem. I don't know. You don't want to have, yeah, I don't know. It's fascinating stuff.
And I'm not an expert on the components of tobacco smoke at all, but are PCBs or were PCBs a component? And I know obviously fish, we know that, and these other exposures, but was that in tobacco smoke as well?
I don't think that, there are definitely, chemicals in the tobacco smoke that could be, and I think kind of what I was trying to get at with that, well, both the root analogy and the sink analogy was that it may not just be one thing. So my idea is it's not just a single, I don't ever think of a chemical as a single smoking gun for causing autism. That's absolutely not. But I think multiple chemicals may be acting through similar pathways. And so it's possible. I mean, I think there are known effects of smoking and hypomethylation on the genome. So that, again, could be acting through that methyl group. I mean, they actually tell smokers to eat a lot of green leafy vegetables, I think, kind of to help counteract those methyl donors that you really need to help counteract the bad effects of smoking.
Yeah, good point. Good point. Okay, thank you. Matt?
Okay, well, I don't see any questions right now. So, Dr. Saul, if you wouldn't mind introducing your colleague Keith, and we'll go ahead and let him talk. And then again, if folks want to ask questions, please feel free to put them into the question box in the webinar toolbar, and we can ask them at the end of Keith's session.
Okay. Great. Yeah, it's my pleasure to introduce Keith Dunaway. I want to call him a doctor because he's almost a doctor. He's very close to getting his PhD. He has actually he's only been in graduate school for a little less than 6 years, but he's been in my lab for 9 years because he started out as a very eager undergraduate volunteer in the lab. And then when he graduated, he wasn't sure what he wanted to do next. He was thinking about graduate school, but wanted some more experience. So I hired him as a junior specialist, which is a technician in the lab, where he worked for a couple of years. And then he was very motivated to go back to graduate school. And so this is the project that he took on, this nice challenging project of trying to figure out the gene-environment interactions of DUP15Q and PCBs. So I will take it over.
Great, thank you, Janine. Is my audio good?
Yeah, but they should probably mute me.
Yes.
You've got my mouse. You want to try to, yeah.
There we go.
Get rid of that. It might help you to get rid of that so you can see your slides.
Great.
Yeah,
sorry, we're just having a little technical difficulties with switching over. All right, here we go. All right, well, I'd like to thank Janine for a very lovely introduction, as well as Alicia, Jill, Joan, and Matt for inviting me to speak here today. And I wanted to start out my talk by talking about Dup15q syndrome. So as Janine had mentioned, that was the most interesting autism spectrum and neurodevelopmental disorder that was found in Michelle Mitchell's study that was published in 2012. And Dup15q symptomatically shows up as hypotonia, short stature, seizures in over 50% of the individuals, as well as an intellectual disability, primarily autism spectrum disorder. Now, the difference between Dup15q and most autism spectrum disorders has to do with Dup15q having a known genetic cause. And that is due to a partial copy of chromosome 15. Now, in normal individuals, chromosome 15, as well as all the other chromosomes, show up as diploid, one copy from your father and one copy from your mother. In Dup15q syndrome, you have the same two copies of each chromosome in every cell in your body, but you also have an extra partial copy of chromosome 15 from your mother. Now, this extra copy of chromosome 15 is a little different than the normal chromosome 15 in that it has the entire P arm as well as just part of the Q arm, 15q11-14. And so the orientation of this third chromosome here is the full P arm, and then 15q11-14, and then due to improper crossing over, another copy of 15q11-14, and then another copy of the P arm. Now, this is important because it changes the ratio of that 15q11-14 region from a 1-to-1 ratio to a 3-to-1 ratio. Now, this area is very important for neuronal development and DNA methylation due to the Prader-Willi imprinting control region controlling a bunch of genes in this area that are important for proper neuronal development. So in the paternal allele, the Prader-Willi imprinting control region is unmethylated, which allows for multiple genes to be turned on, including SNRP-N. Whereas the maternal allele, you have the silencing of the Prader-Willi imprinting control region, and that allows for full copies of UBE3A to be produced that aren't serically hindered. So in the paternal allele, you have, in this example, SNRP-N turned on. It transcribes SNRP-N, SNORD116, SNORD115, and then it goes through and hits the UBE3A, as well as produces an antisense, which diminishes the UBE3A contribution from the paternal allele. When it's silenced, UBE3A is allowed to go fully and produce UBE3A in your cells. Now, Michelle, back in 2012, she found PCB95 to be significantly enriched in Dup15q brain samples, and she also did some line 1 pyrosequencing. Janine had mentioned dot blot, which was one of the first ways to look at global DNA methylation. After that was line 1 pyrosequencing, which is a really great tool to use pyrosequencing to look at the repetitive region of line 1 and have that as a proxy for global DNA methylation. And what she found was a significant decrease in methylation for Dup15q compared to controls. So that was kind of the jumping off point for me. But I wanted to look at every cytosine in the genome. And in order to do that, I utilized whole genome bisulfite sequencing. So the way WGBS works is a kind of brief overview, is that I isolated DNA from brains and then bisulfite converted them. And this process essentially just lets you know which cytosines are methylated and unmethylated, because the cytosines that were unmethylated changed to uracils and then thymines through PCR, whereas the methylation protects from this conversion. And after you put all that DNA onto a sequencer, you get back millions and millions of sequences that you can then align to the genome and determine with base pair resolution what the methylation is. So I sequenced, oops, sorry about that. There There we go. So I sequenced multiple different types of samples to get at what was really going on with the methylation, both globally as well as at the Prader-Willi imprinting control region and the 15q11 to 14. Dup15q has extra DNA as well as a 3-to-1 ratio of maternal to paternal contribution, or 75% maternal contribution. So I sequenced Prader-Willi syndrome, which has 100% maternal contribution, and its inverse Angelman syndrome, which has 0% maternal contribution. And those are due to deletions on their respective paternal and maternal alleles in the Prader-Willi imprinting control region, or that entire region of 15q11 to 14 in general. We also were very interested in looking at the possibility that just having DNA in general, a vast increase of DNA, can give you a decrease of overall methylation due to just having a bunch more methyl receptors. And we used Down syndrome as proxy to that. And Down syndrome is actually a really good proxy to Dup15q because it's caused by trisomy 21. And chromosome 21, the size of that chromosome is roughly the same size as the extra chromosome in Dup15q. On top of that, Down syndrome individuals have neurodevelopmental disorders, so it also gives a more phenotype closer to Dup15q than controls. And lastly, we sequenced just idiopathic autisms, individuals that had no known genetic cause for their autism, as well as age and sex match controls for these. Now, one of the first things I wanted to know was, did we recapitulate the results from pyrosequencing in line 1? And I actually had to do some custom coding for this because normally with whole genome bisulfite sequencing, you don't actually look at the repetitive regions because you can't uniquely align reads. And so it's a bioinformatic nightmare. But I was able to write custom code, it's now available for everyone to use, to take the whole genome bisulfite sequencing data, look at the sequences that were line 1 sequences, and determine if the methylation there was significantly different in my experiment. So pyrosequencing is on the left here. Whole genome bisulfite sequencing is on the right, and as you can see, both have showed this decrease in methylation. It's important to note that with whole genome bisulfite sequencing, overall, we did get an increase in methylation in line 1 compared to pyrosequencing, and that's most likely due to the fact that we're actually able to assay more of the line 1 elements in the genome using whole genome bisulfite sequencing. With pyrosequencing, you actually are restricting your DNA that you're able to assay because you're using 3 sets of primers, a forward primer, a reverse primer, and a sequencing primer to look at your line 1. And that's roughly about 60 bases that has to be unique. Whereas with whole genome bisulfite sequencing, you're looking at about 30 bases. So the whole genome bisulfite sequencing was not only able to look at line 1 sequencing, but all CPGs within the genome. And it turns out that line 1 was a good proxy. We do see a decrease in Dup15 methylation genome-wide, looking at all CPGs. And so we wanted to also answer the question, is this methylation change due to only methylation on chromosome 15 being significantly hypomethylated, or do you see a genome-wide hypomethylation? And so to answer this, we looked at every other chromosome besides chromosome 15 in all of our samples, and it turns out that even though Prader-Willi and Angelman have difference in methylation in the Prader-Willi imprinting control region, just like Dup15, genome-wide, the only samples that were hypomethylated were Dup15. So speaking of the Prader-Willi imprinting control region, I just wanted to show this as a DNA methylation graph across the Prader-Willi ICR, as well as a hypomethylated cluster around there. So each one of these lines represents an average of all of the samples of the particular conditions that the colors are representing here. All of these are normalized to the controls, which is just this gray bar that goes across the middle here. And the first thing you'll notice is that Prader-Willi is in pink, and it is significantly hypomethylated here, whereas Angelman syndrome patients are hypermethylated. And Dup15 is about halfway in between your control and your Prader-Willi. And this is exactly what we would expect if we're looking at this from a dosage standpoint. Prader-Willi is 100% maternal, Dup15 is 75%, Angelman is 0%, and so you're kind of seeing that gradient across this region. Down syndrome and idiopathic samples don't really have a significant difference in this region, and that was also expected. But will also notice that you have these spikes at different areas throughout this region. And those spikes are actually due to CPG islands that are differentially methylated. So your methylation and what it does actually changes depending on what the context is of it. So in the Prader-Willi imprinting control region, the methylation reverses compared to your global methylation across this region. So if you have methylation that is increased in a CPG island that's at the start of a gene, odds are that gene will be silenced or at least decreased in transcription. But if you have methylation throughout the gene body, that can actually increase your chances of having that gene being transcribed. So this slide just shows some statistics from the last slide. As you can see, the cluster is significantly hypomethylated in Dup15q, as well as Prader-Willi, and hypermethylated in Angelman, whereas the down in idiopathic had no change in methylation. And the inverse is true for the Prader-Willi imprinting control region. And this just goes to further hit home that point that if you're looking at methylation that are concentrated in a CPG island, you can have an inverse effect than if you look at larger scale methylation with CPG spread out. Now, as Janine had mentioned earlier, PCBs were an interesting cofactor in this Dup15 experiment. And so we know that PCBs, in particular PCB95, has been linked to hypomethylation. And so we saw that there was a significant hypomethylation in Dup15q, but there wasn't in any of the other genomic autism samples that were affected by changes in this region. So we wanted to get at the question, is this hypomethylation really due to Dup15q having this extra copy of the maternal chromosome 15 Q11 to 14, or is it due to having an increase in PCB95 exposure? And so in order to answer this question, we designed this experiment to look at short-term and long-term exposure of PCB and 15M in a cell culture model. So we use SHSY5Y as a neuroblastoma cell culture model. It's great because unlike most cell cultures, it's primarily diploid. It does have a few copy numbers, which I'll show differences, which I'll show in the next slide. But we also have a great model for 15M in the SH cells as well. So our design was to look at SH cells with and without PCB95 exposure, and then with and without 15M for short-term and then long-term, and see the overall methylation and transcriptional effects of this. But we hit a bit of a snag, which ended up being fortuitous, and that was found out only through a careful analysis of our whole genome bisulfite sequencing data. Normally with these methylation assays, you don't get a whole lot of data that you can do other stuff with. For instance, with pyrosequencing of line 1, you can't get all the other cytosines in the genome, but you also can't detect copy number variation. Same with reduced representation bisulfite sequencing or the other sequencing methods for looking at DNA methylation, or dot plots. So you'd have to do another experiment to get at that data. But with whole genome bisulfite sequencing, we're able to do it with the same data set. So a normal SHSY5Y cell line has, as I said, a couple of extra copies of parts of chromosomes, as well as a few deletions here and there. But for the most part, it's a diploid, which is much better than a lot of the other cell cultures, cell lines out there. And so I took the whole genome bisulfite sequencing data, binned it into 5,000 bases non-overlapping, and essentially looked at copy number variation and found in our cell line this increase in copy number pretty much where we expected it to be, which is a very good thing. So every dot here represents 5,000 bases. The kind of salmon and gold color there represents control brains and SH cells, whereas the other colors represent SH15M, and you see this increase from 2x to 3 and 4x across this region. Now, I didn't just look at chromosome 15, I looked at every other chromosome, and I found an interesting result in chromosome 22. And it turns out that some of our cell lines, the long-term exposure ones with 15M, had an extra copy of part of chromosome 22, and that's shown here. So the SH cell line is represented again as kind of this goldish color. The SH15M is represented as a darker green, and it follows the normal expected for SH cell line copy number to be 2x across chromosome 22, goes down to 1x at the 38 million mark, and back up to 2x at the 44 million mark, indicating a known deletion in one of the chromosomes in SHSY5Y cell line. But our long-term exposure with 15M shows this increase to 3x, and then down to 2x, and then back up to 3x, indicating that the good copy, the full intact copy of chromosome 22 was duplicated starting at the 33 million mark. So that led us to change our experimental design for cell culture to what is shown here, what was in the paper. We still start out with SH with and without PCB95 exposure, and with and without 15M to give it both an environmental and a genetic hit. And then during this 40-day exposure for long-term, a duplication of chromosome 22 happened in the 15M cell line, which then propagated to single-cell cloning. And so for our long-term treatment, we have SH with and without PCB95 exposure, and then with and without 15M plus a copy of chromosome 22. So this led us to a 1-hit, 2-hit, or 3-hit model to look at multiple epigenetic and genetic hits on the genome, or sorry, multiple environmental and genetic hits on the genome, and what the epigenetics were resulting from that. So this is the results from whole genome bisulfite sequencing from that experiment. And as you can see, our short-term and long-term SH cell lines, the short-term represented in green and the long-term represented in purple, have equivalent levels of methylation. But if we look at the 15M and 15M Dup 22, we see a significant decrease here with and without PCB95 exposure. We do also see a decrease in methylation with PCB95 exposure, both in short-term and long-term, but we don't see a significant difference here between short-term and long-term that both had PCB95 exposure, indicating that while the targets may be affected more significantly for long-term PCB95 exposure, we don't see a global change. So with whole genome by sulfide sequencing, I was able to delve deeper than just looking globally. I was able to look at each individual gene and run a gene ontology for genes that were found to be significantly methylated or like increase or decrease in methylation. And I found a increased effect due to the number of hits that the genome had on pathways that were very vital to proper neuronal development. So in 15M, as you can see here, we had some hits in signaling and secretion that were important for neuronal development. But if we add PCB exposure just in the short term, we have a more striking effect on a 15M cell line. And then if we look at 15M with DUT22, we see an even further increase of effect, but the most striking effect is with all three hits, 15M, DUT22, and PCB95. So taking this to a transcriptional look, my colleague and co-first author, Dr. Shahrul Islam, ran a Fluidigm 4848 to look at the transcription effects on the short-term and long-term exposures to PCB95. And this is a pretty complicated representation here, but you can see an overall pattern where the short-term has an upregulation, but the long-term has a downregulation, indicating that there's a time component to these exposures on the transcription levels. So when we looked at this from a molecular standpoint, looking at UBE3A, which is one of the genes that's in 15q11 to 14, we see a significant difference when you have 15M versus a control, and when you have PCB95 exposure. We also have a significant decrease in ring 1B when you have the 15M, indicating and actually confirming that there is a differential effect with having an increase in UBA, meaning leading to a decrease in ring 1B in these cells. And this led us to our model and kind of conclusion from the paper. In a normal individual, you have a normal amount of UBE3A being produced, which leads to a normal amount of ring 1B, and that leads to normal H2AZ with acetylation and ubiquitination, and everything's looking fine with a normal amount of synaptic gene transcription and DNA methylation. However, in 215Q samples, you have this increase in UBE3A, which leads to a decrease in ring 1B, leading to fewer ubiquitination in the H2AZs, and that leads to a decrease in DNA methylation across the synaptic genes. And with that, I'd like to stop here and thank everyone that was involved in this project. And here's just a short list of the LaSalle Lab and our collaborators, and I guess I'll join Janine in taking any of your questions that you may have. Thank you.
Great.
Yes. Oh. And this is just a picture of the lab. There we go, from Halloween. Thank you.
Great. Thank you very much, Keith, Janine. And it looks like you guys have some fun in your lab besides doing some really important work. So I actually don't see any questions. Again, really quickly give folks a chance here. If you'd like to ask a question, put a question in the question box, or you can also enter into the chat box as well if you find that easier. Oh wait, okay. We do have a question that's come in here, so I'll go ahead and ask you that. The question is, so what mechanisms do you know? So understanding the importance of hypomethylation in development. are there things that can be done to address that in terms of improving health, mental development of the brain, neurodevelopment, those sorts of things? As you think forward in your research, are there interventions that you can see that could help address these kinds of hypomethylation issues?
Yeah, great question. I think the simplest one is to take prenatal vitamins, but the key is you got to do that early before before pregnancy. So that's hard, to tell someone who's already pregnant, already has a child with autism. And unfortunately, it seems to be best done early. At least that's from the epidemiology data, to have those extra methyl donors. But I think later on, certainly having a balanced diet. I mean, I think a lot of kids with autism sometimes, from my understanding, is they have struggled to get a balanced diet in there. So there have been some trials done on children with autism and folate that were not, didn't show much. So I think the window is really, unfortunately, very early. But again, I think there's a lot of possibility. I mean, one of the things we have going on in the lab, and is not necessarily a treatment, but what we like to do is be able to have biomarkers of differences in methylation from birth. So we're doing this as part of a prospective study called MARBLES, that's Irva Hertz-Picciotto work, and we can get placenta and cord blood from birth from high-risk families with autism. And so the idea is to try to develop a panel of methylation biomarkers, and again, there could be both genetic and environmental reasons for getting to these same genes that could be hypomethylated. And by knowing at birth if a child has a higher risk for autism, you could probably start earlier on some of the behavioral interventions that do seem to work, and work better the earlier you start them.
Hey, Dr. LaSalle, this is Jill. I'm just going to squeeze in a quick question. I know that we have some more that have come in, but we talked a lot today about imprinted genes. And the methylation on those particular genes is of vital importance, actually very, very early. Again, going back to the germline idea, very, very early in the sperm and egg. And I'm not sure that any environmental contributions can benefit the expression of those genes. It might be just too late if the errors are imposed in those genes early on. Do you have any thoughts about that?
Yeah, I mean, we think a lot about when these patterns occur in the oocyte. It's interesting because the placenta seems to reflect the methylation patterns we're seeing in placenta reflect really the early life patterns that are there in the oocyte and the early embryo. And that's when the imprints are getting set up, but it's also potentially when environmental exposures could leave their own type of imprints on the epigenome. And so I guess that's part of the goal we're trying to find with some of these epigenetic biomarkers. Could we find biomarkers of specific exposures, of general exposures, that would be like, I mean, I guess the holy grail, because I know Prader-Willi and Angelman syndrome were actually diagnosed in the clinic by a methylation change. The methylation, that you could see on some of Keith's graphs. So I guess that's the idea. Could we have an early diagnosis? I think it's going to be more than just one, but if we have a panel of 10 or 20 of these that have methylation changes that could have, again, both genetic and environmental causes of getting to those changes, but could we have biomarkers of these alterations, similar to what you see in diagnosing Prader-Willi and Angelman by the imprint mark?
Right. So actually, the next question I have here from some of the listeners, some of the attendees, is related to just what you're talking about diagnostics. The question is around the idea of if you were interested to know whether or not hypomethylation potentially plays a part in one individual's autism diagnosis or other sorts of associated disorders, are there available diagnostic tests that one could go and get that you're aware of to be able to answer a question like that? That if you're a family or an individual out there in the community who wanted to understand a potential role for hypomethylation, is there something that can be ordered through a physician or otherwise to get an answer like that?
I think there are some tests of metabolites, and again, I think Jill James is really the expert on this because she's really gone after the metabolic aspects. So the ratio of like SAM to SA can tell you, your methyl donor. Yeah, so I think, and I just saw a paper by that group where they actually had a computer learning approach to finding the 5 best diagnostic markers of, and one of them actually was global DNA methylation of their 5 metabolic markers. But I think some of the other metabolites, I don't know if they're standard clinical tests, but again, I think that they're probably there in the near future. There may be some metabolites that could be measured that could tell you if you're at risk for hypomethylation.
Great. And so, given the focus that's gone into, or especially in this talk around 15q, we do have a question from a parent here of an individual, of a 10-year-old who has been diagnosed with interstitial Dup 15q syndrome, and they were curious to know whether or not you were aware of pharmaceutical therapeutics that could. Well, the specific question is around pharmaceutical therapeutics that could target improved methylation. I know you've kind of addressed this in the past, so I'll broaden that to say, as you've continued to investigate Dup 15q and understand a little bit more about the underlying mechanism, both from the methylation but then the genes involved,
Mm-hmm.
do you see the potential for interventions, therapeutics that could both address methylation or potentially downstream effects of that hypomethylation due to some of the resulting biological changes?
Yeah, it's a great question. Yeah, great question. I mean, so what we didn't have time to go into, but in our paper, I mean, what Keith did, which I thought was great, is we took the list of genes. So there was a long list of genes that came out of the study that Keith presented. And we compared that to a list of the druggable gene databases, like DGB. And there's a whole supplemental table in our paper showing those genes. And yeah, with the hope that, we're not saying that these will, cure your child, but there's a list of existing drugs on this that affect a lot of, because we had a lot of neurotransmitter receptors, the GABA receptors, the glutamate receptors, serotonin receptors, they all have many drugs that have been developed against them. So, even bringing that list into your, someone who specializes in controlling your child's seizure potentially could help. I mean, again, we're not saying that we have a treatment for this disorder from this, but the idea was we would share our database, the list of genes and drugs in our paper, if it could be useful to clinicians and parents who want to talk to their clinicians about it.
Okay.
129 drugs. So probably too many to try at once.
Exactly. Well, I think the exciting news from that front is that there are ideas bubbling up that, in getting this deep biological understanding that your work and others is generating, it's leading to new hypotheses that ideally will lead to new treatments down the line. So I have another question here from Sarah Cole. My child has autism and will soon get genetic testing at Stanford. Is it important to let the geneticist know that I, the mother, did eat whitefish in the Upper Peninsula of Michigan and perch in Western Michigan for some frequency at the late 80s, 90s? So again, even just to broaden that out, in sort of helping physicians think about and help provide the right diagnosis, how useful is it to share some of these thoughts around potential environmental impacts that you yourself might be exposed to? Do you have any thoughts around that?
Yeah, I think, at the same time a physician is taking a family history that you would for genetics, I mean, or a family, you're drawing out a pedigree, what other, family members may have autism to look for genetics. I think also if you suspect, even if it's just a suspicion, something, unusual in your environment could have played a role, it's something that they could add to the picture. When you do the genetic testing, they're not going to be testing for PCBs, but it's something that they could keep in mind. Well, if we don't find a genetic cause, maybe it's more of a polymorphism. And the challenge in genetics, I think, is it's really hard because what the geneticists do is, and rightly, is to look for mutations that are in the child that are not in the parents. Right. So, and because that would be a smoking gun. You can, say this gene is likely to cause the autism in your child. But when there is a mutation that's also in the parent, in one of the two parents, it could be that that on its own wasn't causing it, but together with other genes or other environmental factors, it could. So I think the more information you can give the person trying to understand the cause of the autism, the better.
Great. Well, I think we'll go ahead and wrap it up here. Again, I want to thank Jean and Keith both for taking the time to share with us the important work that both of you are doing. And I think really fantastic work in really highlighting the interactions of genes and environment and sort of this complex interaction that eventually underlies a lot of the biological variation that we see across all of us, including and leading into disorders and conditions like autism. So again, I appreciate that. Alicia, Jill, any other concluding words you'd like to add before we end?
I thought that was very, very complicated information. It's very dense. I know that always we have a mix of people who listen to our webinars, some of whom are very sophisticated researchers and some of whom are just, ordinary moms and dads. And as I noted in the beginning, we do send out a link to a recording of all of our webinars. And this link will go out probably tomorrow. And what I usually have to do is go through the webinars slowly, right, to really put the pieces together. And I encourage others to do that.
This is Alicia. So I just wanted to point out to everyone that a lot of these findings were kind of their predecessors, or the reason they were able to study them was through research done in the brain tissue of people with autism and people with duplications of chromosome 15 or mutations in chromosome 15 in autism. So I think I was really struck that in these people, it had been previously thought by geneticists that the diagnosis was 100% genetically predetermined, right? So we've moved away from the term single gene autism because geneticists now, at least totally acknowledge that it's not just the action of one gene, there's probably other genes involved. But people with this type of autism were in fact thought to have, predominantly a genetic or a single gene influence on diagnosis. Through Dr. LaSalle's and or soon to be Dr. Dunaway's research, it's strikingly not true that environmental factors play a huge role in autism diagnosis, or could potentially play a role in autism diagnosis in individuals with chromosomal mutations that were previously thought to be the only cause of autism. So the balance used to be, well, in certain cases it's all genetics, and in certain cases it's all genetics and environmental. And I think because of this work, it's really, it's really kind of called into question that. But I do want to stress that people with it's important to study people with chromosomal, known genetic mutations. Either whether they're part of a chromosome or whether they're, single gene mutations that are grouped together, because it does allow us to understand different types of autism. As Dr. LaSalle mentioned, there's a very high rate of seizures and lots of medical issues in people with these mutations, and it's very important to understand, to use genetics to understand different underlying behaviors when we're trying to develop particular therapies. So just to wrap it up, I want to urge everyone who watched the webinar today to go on to takesbrains.org to register for this program. It does not mean you're obligated to donate your brain after you die or donate anyone else's brain after you die. It just means that you want to learn more about the program, and we have a quarterly newsletter, and we announce webinar series like this as well, for you to get more information about it. So also, just like Matt and Jill said, thank you guys so much for being on the webinar today, and for staying until the very end.
Thanks, everyone.
Great. Thanks.
All right,
Yeah, thank you.
signing off.
Thank you.
Bye-bye.